Okay, so my name is Lukesh, and I will be going over the second part of nervous system. What is nervous system? Basically, it's a communication system. It controls everything that goes within your body and respond to everything that you experience through different simulations around your environment and within your body. Okay, so the second part of the nervous system goes over ions and channels, different kind of ions and channels that we need and we have in our nervous system, different kind of potential, gradient and action potential, and how those potentials move from one part of the neuron to the other part of the neuron. Synapses, neurotransmitters, receptors, neural pools, and processing. Not a lot of things to cover, but still not too less. All right, let's directly just dive into it. First thing first, changes in resting membrane potential. How do we change the resting membrane potential? But why do we do it in the first place? What is it? And second question is, what is it? So let's talk from the beginning. Let me try to explain it from the beginning. Resting membrane potential is nothing. It's the potential of your neuron in this case, or the polarity of your neuron when it's in the resting state. And usually it sits at minus negative 70 millivolt. So emitting potential results from ion separation by cell membrane. So when you have different kind of ions on each side of the membrane, that leads to creation of polarity or potential. Changes in membrane permeability cause ions to cross membrane down the electrochemical gradient, and polarity is affected. And I hope we are clear with this part because we covered everything in the other lecture, part one. So I'll be skipping through these first few topics which we have already covered, and then we'll talk more about the new content. Depolarization is nothing is a decrease in membrane polarity potential, less negative as the membrane loses its polarity. Hyperpolarization is nothing is an increase going towards more negative. Okay, that's what they mean when they say more increase. Increase in sense it's getting more negative. That is more from minus negative 70 to negative 85, negative 90, negative 80, negative 75, something like that. Okay, changes in resting membrane potential. Okay, let me make this a little bigger. This is good. Okay, and we can just sit over here. Perfect. So neurons use deviation of resting membrane potential to signal and send signals among different neurons. It's like if I have, it's for example, if these are two neurons and they want to talk to each other, so this one will send electrical signals, will release neurotransmitters, and this will receive and send the signal forward. So, so for this one to send an electrical signal, need to change the membrane polarity, need to change the resting membrane potential, and that's how it's going to conduct the impulse. Signal can be of two types, gradient potential and action potential. So we also covered these two topics in the other lecture, but we'll go briefly over these. Gradient potential. It's a short-lived, localized change in polarity of varying amplitude and attenuating as distance travel increases. It's a short version, it's a smaller version of action potential, in other words. It's just my understanding. You don't have to put it that way, but just to understand it in our own in our own brains, that it's short distance and it's more localized. It's not like action potential, which once started has to finish. Okay, and it's of varying amplitude, but our action potential is of single and it's always same. And attenuating means it decays as the distance increases. The gradient potential decays. Okay. Whereas action potential is a self-propagating change in polarity of fixed amplitude that travels long distance with high fidelity. That it keeps renewing itself, very self-renewal. It's like a self-start engine. So here the first picture is the gradient potential and the second is action potential. Action potential always looks this way, whereas gradient potential could go up, go negative, go positive, could go negative, positive, negative, and it could reach the threshold or could not reach the threshold. Okay. Let's dive a little more into the gradient potential. The stimulus strength dictates the voltage change and how far it travels. If the stimulus is strong or it's not that strong, that decides If it's gonna go towards the threshold, or that means it's gonna depolarize, or it's gonna go away from the threshold, that it's gonna hyperpolarize. Okay, stimulus causes gated ion channels to open, leading to depolarization or hyperpolarization that can spread by decay over the distance. These are the two keywords for this what do we call it? For the graded potential. Okay, receptor potentials. Okay, so graded potential is of two different types. First is receptor potential, and second is postsynaptic potential. Receptor potential, for example, is the sensory neuron in my finger. When I experience that something is touching it, if it's not strong enough, then I won't, then the sensory neurons will not send a signal. But if there is like the simulation is strong enough, then I will receive the sensory stimulation. For example, we have bacteria sitting on our hand all the time, but do we sense their presence? No. Why? Because the stimulus that we receive from them is not good enough, it's not strong enough to get to the threshold, to reach the threshold. That's why we do not receive it. And that's another example of receptor potential or generator potential. These are the graded potential in receptor of sensory neurons. Remember that keyword. It's the receptors. It's the graded potential in the receptors of sensory neurons. Okay. The second one is postsynaptic potential. It's the neurode graded potential. In other words, the way two different neurons communicate between each other. That's what we call postsynaptic potential. I'll go over the word postsynapsis very soon, but for now, just know it. And that's your diagram explaining the same thing. And let's move on. Action potential. Let's talk about action potential. These are the main neural signal or neuronal signal, however you want to call it, which runs over a longer distance, also known as nerve impulses. They are unique to neurons and muscles. The quick inversion of membrane polarity, that is 100 millivolt difference. Why they're trying to explain is like it starts from minus 70, goes up to plus 30. That's a difference of negative 70 to 0. That's 70. And from 0 to 30, that's another 30. That means 70 and 30. That's a difference of 100 millivolt. Okay. Does not decay over distance due to constant renewal. And at the end of this lecture, if we get some time, I will play another video, or I'll leave you a link which you could go and check out more in-depth understanding of action potentials. Okay, these are generated by successive opening and closing of different voltage gated channels, creating ion flux. That's how action potentials uh, runs from one point to a different point. It works on the mechanism called all or nothing. So action potential either will start and finish or will not occur at all. So all or nothing, it depended on whether the change in the potential was enough or was strong enough to reach the threshold. If it reaches the threshold, all sodium channels will open. All ion gated sodium voltage gated ion channels will open, and the sodium will rush in, and that will lead to the initiation of an action potential. Okay. Next, voltage gated channels of the action potential. These are the following gated channels. We already covered this part, this part, this part, this part, and we also covered this part too. I'm making this video for my nursing students or my classmates, so won't be going over all this because we already covered it. Okay, I don't think we covered. Okay, we'll start from here. Action potential propagation. Action potential has to be transmitted down an entire axon all the way to the end of axon terminals. Yes. Sodium influx in the gates in one area causes local change in the voltage causing opening of adjacent. It's like once you have that influx of sodium, those sodium ions repel each other and will open up the second one. Then the same thing will happen to the next one, the same and the same. And we'll keep opening the adjacent channels like a domino effect. You can see the same thing in the picture. Action potential propagation. Action potential only goes in one direction. So remember that. It only goes in one direction. Sodium channel close to the action potential origin are still inactivated, so cannot generate action potential. And I think you guys know it because we watched the video that explains why it doesn't open. Because the one on the back side, let's say there are some channels on the right side and the left side. The right side is the direction in which action potential is actually moving, and the left side is where it was originated from. So if I show you a picture, and I hope that will make more clear. Okay, I'm looking for something here. I can't seem to find. Okay, let's use this. Okay, we use this one. So the red cap is, you know what? Let's just use this picture. So the blue part is representing where the action potential is starting from, and the pink is where it has to go. So if it starts from the cap here, the blue cap, that's the axon hillock. Why cannot the axon or the action potential moves backwards? Why it only goes in one direction? Because the channels on the back side, on the opposite side, from the direction of its origination, goes into hyperpolarization because the potassium channel starts opening and starts leaking out the potassium. That's why, okay? So it's only one direction, and remember, it's always one direction. Once started, an action potential self-propagates from one voltage channel to another one. In non-myelinated axons, adjacent segments of the membrane successfully depolarize and repolarize, okay? They will keep repolarizing and depolarizing. In myelinated axons, these propagate in a saltatory manner, jumping from one section to the next. I'll show you the picture here. This is continuous conduction. It's a slow process, and this is saltatory conduction, which is quite fast. It is many, if it's faster by many folds, okay? And what do we have here? Myelin sheath. That means it saves, it prevents, or it insulates, or it protects the loss of ions, which happens in case of non-myelinated fibers. Action potential originate in axon hillocks and can only happen in axons. So remember that. Action potential only happen in axons. Okay. Action potential and the speed of conduction vary between neurons. Larger axons usually less resistant to local electrical flow and allowing faster impulse. It's like if it's a bigger bottle, if it's this big, if the lumen or the opening of it, I'll show you the opening. This part of it is this thick. It allows more space and more space for adequate flow of ions. Whereas if it's thinner, it doesn't do that. So myelinated fibers transmit impulses faster than as the action potential jumps from one myelinated area to the next, also known as saltatory conduction. You can look at this picture here. Stimulus is starting here. That's how it moves. Okay. What is saltatory conduction anyways? It's nothing. It's like you have your channels here. So, okay. Let me see. So we have our channels here, and this blue cap represents one channel, the pink cap represents another channel. So in between we have a myelin sheath. So the exchange will happen here, and the ions will travel inside, and the next ion exchange will happen here. Okay, so it allows for impulse transmission 30 times faster. So for example, if the first transmission happening in the what do we call them? Non-myelinated fibers is like 10 running at 10 miles per hour, it's going to be 300 miles per hour, okay? Then the non-myelinated fibers, myelin insulates, reducing charge loss, and voltage-gated channels in gaps between myelin sheets regenerate action potential. So signal jumps from one gap to the next, less ATP required and much faster the process. Okay, sodium channels impairment. What could impair these sodium channels? Those channels are pretty important. Action potential impulse propagation can be impaired by chemicals. Local anesthetics like lidocaine blocks sodium-gated sodium channels. Oh, wait, sorry. Local anesthetics like lidocaine blocks voltage-gated sodium channels. That's why we do not feel any sensation because lidocaine blocks the sodium channels, so you do not receive or send any stimulation. This is kind of medication that we use when we have to perform any kind of like local surgery sort of thing. Pufferfish toxin, just okay, so what is pufferfish toxin? Pterodotoxin is a potent, it's like a really, really strong agonist. So what happens? This toxin replaces sodium and it takes over that. Okay, changes in blood, changes to blood circulation or oxygen delivery can affect neuron functioning. So how much nutrition and how much oxygen is your neuron getting, that could change or affect the sodium channels or can affect the neuron functioning. For example, cold fingers get numb because they're not sending enough neuron signals, or you lose the sensation. Okay, arm or foot goes to sleep because when we're sitting in one position, we cut the flow of blood to that part. Okay, okay. Stimulus intensity and nerve impulse. In a given neuron, action potential has have set amplitude regardless of stimulus. That means they only go from negative 55 to plus 30, come back to negative 70, 80, and go back to the same extent. The amplitude stays same in all kind of action potential. Then how do we feel? Let's say that one stimulus is strong and other one is not. For example, if I feel this against my hand, and if I feel this against my hand, how do I feel the difference? I know there's some water leaked out. It is not the strength of the signal, it's rather the frequency, how faster the action potentials are being sent. Strength of stimulus encoded by frequency of action potential. The stronger the stimulus, the higher the frequency. Okay, remember that. And I can show you the same thing in the textbook if you're interested, so I can give you a better example of it. Just gonna take a second here. Okay, okay. Hmm. Okay, let's see. So, if we look over here, so how can the CNS determine whether a particular stimulus is intense or weak? For example, how can you tell the difference between a quiet hum and a loud roar? The answer is quite simple. Strong stimuli generate nerve impulses more often in a given time interval than a weak stimuli. Stimulus intensity is coded by the number of impulses per second, that is, by the frequency of action potential, rather than increases in the strength of an individual action potential. Hope that makes sense. So we could say this one is not as strong impulse as this one. Say that? I hope you understand. So every second, it's so close and they have so much of distance. The time is on this axis and the memory potential is on this axis. But they all are touching plus 30. Hope that makes sense. Now let's go back. Refractory period. Okay. It's nothing. It's the time during which another action potential cannot be generated in neurons because of voltage gated channels. Huh. I'll show you the picture, and that picture explains it pretty nicely. Two phases, absolute and relative. What is absolute? Absolute refractive period starts when sodium channels open until they reset completely. They causes action potential to be all or none event, causes action potential to be one direction only, the sodium channels. So which channels are responsible for the absolute refractive period? Your sodium channels. So voltage gated sodium channels. What about relative refractive period? Right after absolute refractive period, some sodium channels are reset, some potassium channels are still open, leading to repolarization or hyperpolarization. Larger difference between memory potential and threshold. Action potential can be generated only by a very strong impulse, which is going to cover that gap between hyperpolarized and just polarized. That is the resting moment potential strength between negative 90 and negative 70. If the stim impulse is that strong, if it can cover that gap, then only you can send a signal in the relative refractive period. But so, in which period can we actually shoot another impulse or shoot another action potential? Is it absolute or relative? It's relative refractive period. Remember that. So this is a picture. Let me show you another picture of the same thing, and that picture explains it way, way better. So once sodium channels open and until they are closed, that's your absolute refractive period. You cannot shoot any action potential. This is relative. Relative starts from here when the sodium potassium channels are almost closing up, but they're not closed yet. And that's the time when we can shoot an action potential, but it has to be a strong stimulation for us to be able to send a signal, okay? All right, so mm, 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 mm. And there's a good example if you want to remember this thing. If you guys just look at this, I don't know if I can highlight it. There we go. It might help you to remember the difference between the absolute and relative refractive period if you know that the word refractory means stubborn or unmanageable. Think of a dog that is refractory, disobedient. If he's absolute refractory, he won't come no matter how loudly you call. But if he's only relatively refractory, he may come, but only if you call loudly enough that he knows you really mean it. Right on. Okay, so that's our refractive periods. Now let's talk about the synapse. What is synapse? The main function of the nervous system is to allow communication between neurons. That's how we are talking. For example, the motor neurons, the upper motor neuron and the lower motor neuron, we need to communicate, right? This is your the axon terminals for the upper motor neuron. This is the dendrites and cell body of your lower motor neuron in the spinal cord. That's where they're gonna connect, right? That's where they're gonna communicate. Sorry. Neurons communicate with each other and with effector cells via synapses. A synapse is the site between presynaptic neuron that conduct impulses towards synapse and a postsynaptic neuron or cell that will be receiving the signal. So one is sending the signal, second is receiving the signal. Shoo. Okay. Most neurons are interneurons. That's the function as they function as both pre and postsynaptic. So what interneurons means that they can either receive or send signals. They can do both of them. They can receive signals in dendrites and the cell body part, and they can send signals through the axon terminal part. Hope that is kind of in the picture that you guys can imagine it. Uh, in PNS, the postsynaptic cell may be a neuron, a muscle cell, or a gland cell. That's the part. So in a PNS, not in the CNS, in the peripheral nervous system, the postsynaptic cell may be a neuron, so it's a neuron communicating with a neuron, or a muscle at a neuromuscular junction, or a gland cell to release something out of it. Okay? All right. Types of synapses. Okay, let's go with types of synapses. Synaptic connections could be either between one axon and an axon and dendrite, axon and cell body, dendrite to dendrite, dendrite to soma or cell body. Let's look into the textbook because they have a nice bigger picture. So these are some dendrites, the cell body. These are axon with their axon terminals. So in this one, the axon terminals are attaching or synapsing with the cell body. They're called exosomatic because axon and soma. Here the axons are communicating or synapsing with the dendrites, so exodendritic. Now here we have two axons, one axon connecting with another axon or communicating with another axon. It's called axonal synapses, okay? Okay, let me just synapses between axon endings of one neuron and the dendrites of other neuron, exodendritic. Those between axon endings of one neuron and the cell body or soma, exosomatic. Less common or far less understood are synapses between axons and axons, between dendrites, dendrodendritic, or between cell bodies and somatodendritic. There are two types of synapses now, electrical and chemical. Let's look at that. We're gonna talk about the first one, the chemical one first, and I think you guys have a pretty good like understanding of this. And I'm not saying you need to have it, but like if you don't, we'll try to understand it here as well, okay? But if you guys remember from your neuromuscular junction, so when our axon releases those neurotransmitters and the junctional folds have those chemically gated ligand channels or the ligand channels, and they receive that chemical called acetylcholine. That's an example of chemical synapse. Okay, synapse. Let's talk about them. Widespread type of synapses that occur using neurotransmitters, which are also chemicals, to transmit nerve impulses are called chemical synapses. So what do we need? We have axon terminal of presynaptic neuron, which has synaptic vesicles or just vesicles. Vesicles are those pouches that contain neurotransmitters, or also known as chemicals, containing neurotransmitters. Then we have fluid-filled synaptic cleft, where neurotransmitters are released. That's a gap between one neuron that's sending the neurotransmitters, and then there's another one that's going to receive it. But the gap in between, it's called synaptic cleft. It's not empty, it's not air, it's fluid-filled when neurotransmitters are released. And then we have receptor region on the postsynaptic neuron or neuron's membrane with specific receptors for neurotransmitter. Okay, electrical impulse translated into a chemical impulse across synapse, then transformed back into electrical impulse. So something triggered. So axon, we had action potential coming down the axon into the axon terminal that leads to the neurotransmitter release. That means the electrical activity changed into chemical activity, and once those axon, the neurotransmitter will attach to the receptor, they will generate new electrical activity. Okay, it's like electrical, chemical, electrical. So electrical impulse translated into chemical signal across synapse, then transferred back into electrical signal. See how sophisticated that is. Unidirectional communication that depends on release, diffusion, and binding of the chemical to its specific receptor. Well, that makes sense. Now, chemical synapse. Action potential arrives at axon terminals of the presynaptic neuron. Voltage getting calcium channels open. Calcium enters axon terminal. Calcium entry causes the neurotransmitter release. Calcium associates with the vesicle causing fusion with the neuron membrane. Neurotransmitter excitosis in the synaptic cleft. Higher action potential frequency causes more higher action potential frequency. So now it's like if it's a bigger stimulus, this is what's going to happen. Higher action potential frequency causes more exocytosis, more prosynaptic effects. And neurotransmitter diffuse in the synaptic cleft, binds receptors on the postsynaptic membrane. Let's look at the picture in the textbook. That's a gigantic picture. So presynaptic neuron, we don't know what this is. Let's imagine it's a postsynaptic neuron. It is actually a postsynaptic neuron. Action potential is being shown by these arrows. We have mitochondria, we have vesicles containing different chemicals or neurotransmitters. Then we have calcium channels. When action potential reaches these calcium channels, they change their shape and open them. Calcium flows in from ECF to ICF, extracellular fluid to intracellular fluid, and then it forces these vesicles to fuse with the nuclear membrane, and as a result, these vesicles release their chemicals. Here on the postsynaptic membrane, we have these chemically gated channels. So these neurotransmitters will diffuse across, attach here, and do whatever they have to do. Okay. Also, they cannot stay here all the time. So what's going to happen? The enzymatic degradation will happen here. So that neurotransmitter could be put back in here. Okay. We're not going to go to step-by-step process because we already covered it in the neuromuscular junction. But if you guys want me to, we'll do that in the next class. Just remember, if you are interested in doing that. Okay. All right. So that's our chemical synapse. What's the fifth step? Neurotransmitter binding open ion channels. So this is called graded potential. So now, whatever happening on the surface of the pre and post synaptic neuron that might or might not lead to a generation of initiation of an action potential is called graded potential. So the receptor protein change shape causes ion channels to open. Remember when I said these are chemically gated ion channels. Even though these are the neurotransmitters which are going to be affecting these channels, what is the main reason? What's the main thing that's going to happen as a result of it? It's like those chemical gated ion channels will either open or close or will let something in or out. Some can be excitatory or inhibitory. That will depend on what kind of ions are traveling through those channels. Okay, some receptor proteins are ion channels. So some of them. I don't know. I don't know about the rest of them. Maybe we'll cover those. Don't worry. So termination of neurotransmitter. Neurotransmitter needs to be eliminated in the absence of signal. So once the signal came, that did its job. Now there's no more signal. What's going to happen to the neurotransmitter? Those neurotransmitters have to be degenerated or degraded. Shortly after release, few milliseconds of the neurotransmitter release, it is taken up by the astrocytes or axon terminals. Okay, is enzymatically degraded and diffuses away from the cleft within milliseconds. But there are some medications like SSRIs. So these are antidepressants called serotonin release, serotonin re, okay, serotonin, norepinephrine reuptake inhibitors. So what are they doing? So serotonin is one of the neurotransmitters, just like acetylcholine that works on muscles. Serotonin makes us happy. In people who are depressed, this is like a finding that the serotonin levels are decreased, or they don't have a lot of serotonin levels in their synaptic cleft to make them happy or to keep them happy. So what these medications do, which are called SNRIs or SSRIs, they inactivate these enzymes. They're going to break these down and put them back in the exon terminals. So if that doesn't happen, these neurotransmitters could stay in the synaptic cleft for a longer period of time, and as a result, it helps. And I don't know a lot how it helps, but it helps for sure. Chemical synapses again, continuing that, sorry. Although action potential transmission is very fast, a limiting factor in signal transmission is the, even though the action potential transmission is very fast, but a factor that slows it down a little bit is synaptic delay. Synaptic delay is minimum time required for transmission across the synapse for neurotransmitter exocytosis, diffusion in the synaptic cleft, action on the receptor, change in postsynaptic cell membrane permeability, and diffusion in the postsynaptic cell. So all these for all these processes to happen after an action potential is already like conducted through an axon, this part delays it. It's called synaptic delay. And if any of them does make sense, we can bring them up. Okay, we can talk about them. All right, that's the summary. That's the picture we just saw in the textbook. Now electrical synapse. So we also have electrical synapses. What are they? So the relatively rare type of synapses where neurons are electrically sorry about that. They're coupled by gap junctions. So it's nothing. It's just these are the channels which are open and which leads to shading of cytoplasm. That's the material within the cell. For a good understanding, let's look at the Plex book. Electrical synapses. Electrical synapses are much less common than chemical synapses. They consist of gap junctions, like those found in between certain other types of body cells. These are channel proteins, also known as connexons, connect the cytoplasm of adjacent neurons and allow ions and small molecules to flow directly from one neuron to the next. It's like they will let the ions to flow directly from one ion to another. So we have an action potential in the first one, leads to those positive ions and negative ions to travel directly into the next neuron. They do not have those neurotransmitters or anything, okay? All right, so these neurons are electrically coupled and transmission across these synapses is very rapid. They do not have the synaptic delay at all. Depending on the nature of the synapse, communication may be unidirectional or bidirectional. This is also true that the communication could be uni or bidirectional in these electrical synapses. Electrical synapses between neurons provide a simple means of synchronizing the activity of all interconnected neurons, that they all work in similar patterns, okay? In adults, electrical synapses are found in regions of the brain responsible for different movements like eye movements, also found in the hippocampus part of the brain, which is responsible for memory and emotions. Electric synapses are far more abundant in embryonic nervous tissue, that they are found in like when our growth is just happening in the embryo, when they permit exchange of guiding cues during early neuronal development, so the neurons can connect properly with one another. As the nervous system develops, chemical synapses replace some electrical synapses and become the vast majority of all synapses. For this reason, we will focus on chemical synapses from now on. So that's why the textbook is focusing on the chemical synapses. Now, postsynaptic potentials. What's happening between an axon terminal and the the dendrites or the cell body? What is happening at the postsynapses part? The binding of neurotransmitter generates graded potentials. We agree with that much, do we? Yes. The amplitude and polarity of graded potential is dictated by three things. So how strong or not so strong the graded potential is going to be depends on uh, three things. How much neurotransmitter is exocytosed, how long neurotransmitter was left in the cleft, and type of ion channel affected. Okay. The graded potential can be excitatory postsynaptic potential, EPSP, excitatory postsynaptic potential, EPSP. So E for excitatory, PS for postsynaptic, and potential for P for potential. inhibitory postsynaptic potential, IPSP. So these are two important terms, and try to remember those because the questions will appear on those topics. And these are the things that affect those. Know these two. Let's talk about the EPSP, the excitatory ones. Neurotransmitter trigger opening of ion channels such as sodium and calcium that causes a local depolarization. So easy way to remember the difference between excitatory and inhibitory is like anything that leads the membrane potential towards depolarization, makes it less negative, it's called excitatory. Anything that leads it to hyperpolarization or more negative, it's called inhibitory. For example, if you look at this picture, the green one is EPSP. It's leading towards negative 55 from negative 70. But if we talk about IPSPs, it's leading towards back to negative. Okay, so the EPSPs can trigger action potential if EPSP is of the threshold strength and reaches axon hillock. These two elements actually need to be there. It's not that if it's only meeting the threshold strength, but also it needs to reach the axon hillock, because that's where the axon will start or initiate the conduction of impulse, or also known as action potential. Once at hillock and if strong enough, voltage getting open, channels open, and action potential is generated, starting nerve impulse. And that this is the answer for your question, Stephanie, when you were asking. Even though they're like touching the threshold strength, why is it like still a maybe? Yes, it's still a maybe because they might or might not reach the axon hillock. This is the part of the axon hillock, okay? Just the beginning. IPSP. Neurotransmitter binding to the receptor opens ion channels to the potassium or chloride, potassium leaking out or potassium or chloride leaking in, causing local depolarization called IPSP. IPSP increases change in voltage necessary to reach increases. So IPSP increases the change that is needed to reach the threshold, and neurons likely to generate an action potential. Okay. Gradient potential summation. Now it's like the resultant of both of those IPSPs and EPSPs will decide whether the action potential will be generated. Also, the other factor is if the resultant or these resultant potentials are reaching, or the ions are actually reaching the axon hillock. Most neurons receive signal from multiple inhibitory and excitatory neurons. Yes, yes. For example, these green ones are EPSPs, the green ones, and the red ones are IPSPs. Okay, so now we have more input from the EPSPs, the excitatory, and less input from the red ones, which are the IPSPs. That's why we see an action potential here. And a single EPSP is not sufficient to generate action potential. That's right. So it could be a statement in your exam. EPSPs and IPSPs are add up, summation, and if EPSPs are dominate and bring membrane potential to threshold, action potential is generated. Whew. Okay, if EPSPs and IPSPs add up, summation, if EPSPs dominate and bring membrane potential to threshold, action potential is generated. In the present that the ions actually reach the action hillock. Okay, gradient potential can summate temporarily or spatially. I don't know how to pronounce it properly, and we'll go with that now. Let's talk about the gradient potential summation. Now, how do we figure out? How do we decide? How do a neuron decide whether to send an action potential or not? It's decided on the basis of few things. Most neurons receive signal from multiple inhibitory or excitatory. For our convenience, I'm just going to call them ISPs and IPSPs and EPSPs. Okay. So if we have multiple of those, so the total sum of all of their input actually decides whether we're going to reach an action potential or not. So let's dive into this topic a little more deeper, and we'll go into the book because there's a nice looking diagram there. So no summation. If there is no summation, then we have temporal summation, spatial summation, and spatial summation of EPSPs and SPSCs. That's it. Promise. Okay. So for example, a situation in which we only have a slow firing presynaptic neuron, which causes EPSPs, that means excitatory part, that are far apart in time. Yes, one now, another one after a minute, another one after a minute. So two EPSPs are separated in time, do not act together because they decay over time, if we remember, and they decay over time and they decay over distance. Threshold is not reached, so no action potential generated. It's like someone calls casually shouting fire every few minutes. Second one is temporal. A rapidly firing presynaptic neuron causes rapidly, remember the word rapidly, that causes they are close in time. Two EPSPs close in time add together, and the word temporal here means time. Summation brings the action initial segment to the threshold and action potential is fired. It's like someone shouting fire, 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 fire quickly over time and over and over. Spatial is like if more than one presynaptic neuron fires at the same time. EPSPs are generated at different locations but at the same time. Two EPSPs at the same time but different locations and add together. Spatial means location. Summation brings the action initial segment to the threshold and action potential is fired. It's like many people shouting fire, fire, fire at the same time. Okay. Now we don't only have EPSPs through coming from different presynaptic neurons. So what happens if we have EPSP and IPSPs? If a presynaptic neuron creates an IPSPs, and it can override the EPSPs created by another neuron, and EPSPs bring the neuron closer to the threshold, and IPSPs bring the neuron away or farther away from the threshold. Together they can nearly cancel out each other. It's like a person shouting fire while other shouts do not fire. And so if one dominates over another, that will decide whether we will have action potential or not. I hope that explains it. That's a temporal, spatial. Now they're talking about the diagram. I need you guys to go through this diagram and try to make sense out of it. And it's the same thing, we just went over it. It's like difference between graded potential and your action potential, and maybe the EPSPs and IPSPs too. Not sure. Neurotransmitters, the integral part of neuronal communication. Neurotransmitters have various chemical structure and effects. Most neurons can make several neurotransmitters released based on stimulation frequency. So if we look at this, most neurons can make several neurotransmitters released based on the stimulation frequency. Okay. Main categories: cholinergic, biogenic, amine, and neuropeptides. Now we'll go over these neurotransmitters. Okay, let's focus on neurotransmitters now. So we have multiple types. I'll show you guys a picture. I'm not sure I'll be able to share it or not, but if I can, I will try my best. Let me try. Let me know, guys, if you can see the picture. And you guys can use this uh, picture to have an easy understanding. I'm not saying this is the best picture, but this is like a shorter version of it. And if it helps, it helps. If it doesn't, then we have a table, a nice looking table in the textbook, and in the textbook that I provided you guys with. Okay. So main categories. What main categories do we have? We have cholinergic, biogenic amines, and neuropeptides. Let's go over them briefly because I'll try my best. Let's see. One of the first neurotransmitters is identified and best understood. It's acetylcholine. It's the one we have at the neuromuscular junction as well. Released at neuromuscular junction and by many ANS neurons as well as some CNS neurons. Binds to nicotinic and muscarinic receptors degraded by an enzyme called acetylcholinesterase, as the name suggests. There we go. That's a picture of a neuromuscular junction there. Then we have amino acids. Amino acids are the building blocks of proteins, so are not evident to play a role as neurotransmitters. Known neurotransmitters are glutamate, glycine, GABA, gamma aminobutyric acid derived from glutamate. So glutamate leads to formation of gamma, and aspartate, typically eliminated by synapse by reuptake and recycling by postsynaptic or presynaptic or the glial cells. Sorry, that's how it's happening. So the GABAs, glutamates, the receptors, and then they're taken back in the presynaptic cells or by glial cells. Examples are glutamate, glycine, GABA, and aspartate. When we have biogenic amines, they are produced enzymatically by amino acid from, so we use amino acids and we use multiple enzymes to make these biogenic amines. For example, catecholamine, One of them is dopamine, norepinephrine, and norepinephrine made from tyrosine. That's the raw material that we use to make these tyrosine. Then endolamines, examples are serotonin and histamine. Serotonin made from tryptophan and histamine made from histidine. Most are recycled by transport back into presynaptic cells, just like the amino acids. Okay, let's highlight that part. Widely used in brain, play roles in emotions, circadian rhythm, that your sleep and wake up cycle, used by some ANS motor neurons, when we talk about norepinephrine. Mental health can be affected by neurotransmitter imbalances. If we have imbalance of these neurotransmitters, they could lead to mental health issues. And I think we have medication to deal with them. Xanax. Not sure how that works. I think Xanax is Lora Xanax alprazolamine. Not sure how that works. We'll look into that. Neuropeptides. Chains of 5 to 30 amino acids. Substance P mediate for pain signals. Endorphins. These are natural opiates which reduce pain. Examples are beta endorphin, dynorphin, or enkephalins. Gut-brain peptides, which regulate digestive processes like somatostatin and cholecystokinin (CCK), usually released with other neurotransmitters, typically not recycled once released. They're just wasted out. Can act as hormones as well. And the one we know is pretty popular is endorphins, the one that we release on exercise. And talk about purines and gases. Purines are nucleic acids used in both PNS and CNS. ATP can act as a neurotransmitter. Okay, remember that. It's adenosine triphosphate. Adenosine inhibitor in brain is the caffeine antagonist of adenosine receptor. So adenosine inhibitor in the brain is caffeine, which acts as antagonist of adenosine receptors. Okay, it works against. Antagonists are against. They're like a villains. Antagonists. Okay. Who's playing the antagonist role in this movie? Who's the villain in this movie? Then we have gastrotransmitters. Gastrotransmitters. Gases are lipid soluble and are synthesized on demand. Example, nitric oxide involved in learning and memory, brain damage in stroke patients, and intestinal muscle relaxation. Hydrogen sulfide gas affects ion channels directly, and then carbon monoxide that affects brain, intestine, and our vascular function. Then we have endocannabinoids. These are lipid-based neurotransmitters, like fat loving. On-demand synthesis, there is no storage. Action terminated by enzymes. Act on CB1 and CB2 cannabinoid receptor that THC binds to. Most common G protein linked receptor in brain, involved in motor learning, memory, appetite, and pain. Won't go into too much in detail. That's a table that could help you remember these. And there's a nice big table in the textbook. Okay, it's pretty huge though, but you can read a few things through this table and make sense out of them. And you can highlight the book or the table or print it, the PDF version of it. Then functional classification. How do we classify the neurons, the neurotransmitter on their basis of their functions? So neurotransmitters are either excitatory or inhibitory effects. Neurotransmitter effect can be excitatory or inhibitory. That is, excitatory means depolarizing and inhibitory means depolarizing, and hyperpolarizing, sorry. Effect determined by the type of the receptor involved. GABA and glycine are usually inhibitory. Glutamine is usually excitatory, but it also depends what kind of the receptor do we have involved in the process. ACH and norepinephrine bind several receptors types with different effects. Acetylcholine is always excitatory at neuromuscular junction in skeletal muscles, but can inhibit cardiac contraction. There we go. And this picture explains the whole thing again. Now, inotropic and metabotropic. Neurotransmitters, receptors. Now we're going to talk about receptors. Neurotransmitters effect can be direct or indirect. Direct action. Neurotransmitter binds to ion channels. Opening them are called inotropic. Membrane potential is directly altered by the ion flux, ACH, and amino acids. Whereas indirect action could be neurotransmitter acts via secondary messenger, also known as metabotropic receptors. Broader, slower, and longer lasting effects. Example are biogenic amines, neuropeptides, and gases. Let's look at the example here. Neurotransmitter binds to metabotropic receptor, the red ones. First messenger release in the synthesis effect. And okay, so this is how it works. So the chemical attaches to the first channel or this first protein, and then they release a step of process. Then they end up eventually they release different proteins which go and attach to a different protein channel that opens or closes under the influence of the first channel. So first thing is first neurotransmitter binds to metabotropic receptor, the neurotransmitters. Then they activate G protein and that binds to the effector protein. So the G protein releases ATP. The second messenger molecules produce activating enzyme that opens this channel. This is the channel that leads to the either ions go in, ions don't go out, depends what kind of effect it's going to have, inhibitory or excitatory. So we have metabotropic and we have inotropic. Remember that. Let's talk about inotropic a little more in depth. These are the common ones that we usually talk about. Channel linked receptors, ligand gated ion channels, if you guys remember the name. The action is immediate, specific, and brief. Excitatory receptors are channels for small cations. Sodium influx contribute most of the depolarization. Inhibitory receptors allows chloride influx or potassium efflux, or hyperpolarization, in other words. Metabotropic. These are G protein linked receptors. Responses are indirect. Well, we know how it's indirect, starting from here, affecting this, and then opening this one. Complex, slow, and durable. Involves integral protein complexes, triggers secondary signals like CAMP3, CAMP or IP3, cause widespread cellular changes. Okay. They open or close ion channels, activate gene expression of protein synthesis, and activate enzymes. Neural integration. Neurons are not working in isolation. Functions are carried by groups of neurons. Neuronal pools. Okay, let's talk about this topic in depth. Okay, so let's go over the neural integration part. Neurons are not working in isolation. They rather work in groups because we got billions and billions of them. Neuronal pools integrate signals and pass it on to produce complex functions. So we have different patterns in which neurons work. One of them is serial processing. We have parallel. We have diverging, converging, parallel, upward charge, and reverberating. So instead of just going over this picture, we'll go over it in the textbook. Okay, patterns of neuronal processing. First is serial. Let's go over the whole thing, and then we'll go over the pictures. And that will explain, because that's how I understood it. See what input processing is both serial and parallel. In serial processing, the input travels along one pathway to a specific destination. In parallel, on the other side, the input travels along several different pathways to be integrated in different CNS regions. Each mode has its unique interventions, but as an information processor, the brain derives its power from its ability to process in parallel, like multitasking kind of thing. So, serial processing. In serial processing, just highlight it for us. Okay, come on, guys, help me. In serial processing, the whole system works in a predictable all or nothing manner. One neuron stimulates the next, which stimulate the next, and so on, eventually causing a specific anticipated response. The most clear-cut examples of serial are spinal reflex. Straight through sensory pathways from receptor to the brain are also examples. Because reflexes are functional units of the nervous system, it is important that you understand them early on. Reflexes are rapid, automatic responses to stimuli in which a particular stimulus always causes the same response. Same stimuli, same response. Reflex activity which produces the simplest behavior is stereotyped and dependable. For example, if you touch a hot object, you jerk your hand away and an object approaching your eye triggers a blink. Reflexes occur over neural pathway called reflex arcs that have five essential components: receptor, sensory, neural, CNS integration center, and motor neuron and effector. Look at the picture. If you were interested, receptor, then we have sensory neuron, integration, center, motor neuron, and effector. Okay, then we have our first parallel processing. Then we have serial, and then we have parallel. In parallel processing, inputs are segregated into many pathways, and there are multiple examples of parallel processing. We'll go over those ones. Diverging circuit, and let's go with the type of circuits we have. We have diverging circuit, in which it has one input and many outputs, and amplifying circuit, so one input and then lots of outputs. Example, a single neuron in the brain can activate 100 or more motor neurons in the spinal cord and thousands of the skeletal muscle fiber. Okay. Then we have converging circuit. What is that about? It's many inputs, one output. A concentrating circuit. For example, different sensory stimuli can elicit the same memory. For example, if we see something and hear about it and think about it. So all those things, all those sensory input can generate a single output, right? Then we have reverberating circuit. It's like signal travels through a chain of neurons, each feeding back to its previous neurons. For an oscillating circuit controls rhythmic activity involved in breathing, sleep-wake cycle, and repetitive motor activities such as walking. Then we have parallel operating circuit, signals that stimulates neurons arranged in parallel arrays that eventually converge on a single output cell. Impulses reach output cell that are at different times, causing a burst of impulse after called an after discharge. Example may be involved in it may be involved in exacting mental processes such as mathematical calculations. So those are the types of neural pools we have, and that's about it. Okay.